Hi, this is Akanksha with Belinda and Elizabeth. We will be going over topic 9.2.5 to 9.2.10 of the IB Biology syllabus, Transport and Angiospermophytes-Transpiration. 9.2.5 Define transpiration. Transpiration is the loss of water vapor through openings from the stomata located on the roots and leaves of plants. 9.2.6 Explain how water is carried by the transpiration stream, including the structure of xylem vessels, transpiration pull, cohesion, adhesion, and evaporation. The leaves absorb sunlight and heat is produced. A humidity gradient is created between the spongy mesophil, which is saturated with water vapor, and the surrounding air. The water vapor will then evaporate and exit through the stomata pore and out into the air. This evaporation of water causes more water to be pulled upwards into the spongy mesophil by a positive pressure known as transpiration. This creates a lower pressure, which through suction pulls the water upwards. Water is transported passively where ATP is not needed. Transpiration is first possible when the mineral ions are actively taken up by the root and water moves into the root through osmosis. This continuous column of water going up from the roots to the leaves is known as the transpiration stream. Water can travel up through the roots and then to the xylem and then the leaves because the water molecules are held together due to cohesion because of the existing hydrogen bonds between water molecules. Water is therefore drawn up the stem by cohesion between water molecules and adhesion to the xylem vessel walls. Water is then transported in the xylem tissue after it goes up to the roots. The xylem continues to transport minerals and water and provides support to the plant. The xylem consists of vessel elements like tracheids and parenchyma cells and fibers. Both cells at maturity are dead and only their cell walls remain. Vessel elements are more efficient at water transport. They are stacked end on end and are typically wider. They have perforations which connect vertical cells and pits which can connect lateral cells. Tracheids are long tapering cells which are connected by pits which transport water.
9.2.7 State that guard cells can regulate transpiration by opening and closing stomata. Guard cells can open and close stomata to regulate transpiration. Stomata are pores in the lower epidermis. Each stomata is formed by two specialized guard cells. During the day, the pores open to allow carbon dioxide to enter for photosynthesis. However, the plant will experience water loss. If the water loss is too severe, the stomata will close. During the night, plants can't photosynthesize, and so the plant closes the pores, thereby conserving water.
9.2.8 State that the plant hormone abscisic acid causes the closing of stomata. The hormone abscisic acid is released in the plant at night when a plant is going through water stress or lack of water. The hormone is a signal for the plant to close the stomata. Dehydration of the mesophyll cell causes the release of abscisic acid, which will stimulate the stoma to close as the plant's system of water retention.
9.2.9 Explain how the abiotic factors light, temperature, wind, and humidity affect the rate of transpiration in a typical terrestrial plant. The greater the rate of transpiration, the greater the water loss from a plant. From this water loss occurs through the stomata, with other surfaces adapted to reduce such losses. Brown and Escombe in 1900 showed that although the stomatal pore is small, the sum of the circumferences of the pores in a leaf effectively mean that with the pores open there is no lower epidermis. There are a few abiotic factors affecting the rate of transpiration. The first is humidity, which is a measure of water vapor in the air and is normally expressed as a percentage. The water vapor forms diffusion shells of changing percent humidity. The steepest gradient is found at the edge of the pore where effectively most water loss occurs. The significance of Brown and Escombe's research now becomes evident. In high humidity, the diffusion gradient is not as steep, and the rate of diffusion is less. In high humidity, the diffusion shells of water vapor from one pore to the next joint, and the steep gradient associated with the edge of the pore is lost. The formation of this boundary layer of high humidity reduces the rate of transpiration. Wind. Moving air reduces the external water vapor concentration such that the gradient between the substomatal airspace and the surrounding air increases. Still, air allows the building up of boundary layers as shown above, and so reduces the rate of transpiration. Temperature. The leaf absorbs light, and some of the light energy is lost as heat. Plants transpire more rapidly at higher temperatures because water evaporates more rapidly as the temperature rises. At 30 degrees Celsius, a leaf may transpire 3 times as fast as it does at 20 degrees Celsius. The effective evaporation is in the substomatal airspace and increases the gradient of water vapor between the substomatal airspace and the surrounding air. Light. The rate of plants transpire is faster in the light than in the dark. This is because light stimulates the opening of the stomata and warming of the leaf. The mechanism of pore opening relates to the guard cell being open. The guard cell absorbs light and then produces ATP in the light-dependent reaction. Then the ATP is used to drive proton pumps that pump out hydrogen plus ions. The inside of the cell becomes more negative. The potassium ions enter the cell, which increases the solute concentration. Lastly, the water moves from the surrounding tissue by osmosis.
9.2.10 Outline 4 adaptations of xerophytes that help reduce transpiration. A xerophyte is a plant that is adapted to dry environments. These adaptations have been made to increase water retention and to conserve water. Xerophytes will live in climates with some form of water stress, including the desert, which is a biome with high temperatures and low precipitation, areas with very high or low altitude that have low precipitation or water predominantly locked as ice or snow, or areas with rapid drainage such as sand dunes. To avoid transpiration, there are several adaptations that a plant may have in order to retain water. Firstly, a plant may have waxy leaves. This is when a plant will have a waxy cuticle on its upper and lower epidermis, trapping water inside the plant cells. If a plant did not have this waxy texture, it would die through water loss because the cellulose cell wall is not capable of effective water retention. Secondly, a plant may have rolled leaves, stomatal pits, or hairs on the epidermis, such as the marram grass leaf. A rolled leaf will create a cocooning effect on one side of the stomata. This puts the stomata in an enclosed space protected from the wind. Stomatal pits are grooves in the leaf, allowing boundary level of humidity to build up, reducing evaporation. The hairs on the inner surface of the leaf will trap water vapor, reducing water loss through the pore of the plant. The sort of lumen created by the roll in the leaf acts as a channel for rainwater to the roots of the plant. Thirdly, the plant may have needles as leaves, such as firs and pines. Basically, these would consist of coniferous plants in taiga forests ranging from Russia to North America. Needles effectively have no lower epidermis, very much like a rolled leaf. This adaptation has occurred because these plants must survive in cold northern climates, in which water is frozen for long periods of time. These plants are therefore in an environment comparable to desert environments where water availability can be scarce. This adaptation means that conifers can be distributed further than just the northern forests to many climates where water stress is, such as the Southeast Asian mountains. The shape of the needle directly reduces the transpiration, and the fleshy stem contains the water in the leaf, which is the main photosynthetic tissue. CAM and C4. CAM, crassulacean acid metabolism, CAM, in plants reduce water loss by opening pores at night and closing them during the day. This is a time-based alteration of biochemistry. At night, carbon dioxide is combined with phosphoenol pyruvic acid, C3, to form oxaloacetic acid, C4. Oxaloacetic acid is changed to malic acid or aspartic acid. This stores the carbon dioxide until required for photosynthesis during the day. During the day, the pore is closed and the malic acid degenerates to PEP, C3, and carbon dioxide. Carbon dioxide is then used in photosynthesis. C4. A C4 compound is temporarily stored in the spongy mesophyll, which lack rubisco for carbon fixation. Carbon dioxide is stored in the mesophyll later by combining with the PEP C3 to form oxaloacetic acid and malic acid, as seen before in the CAM plants. This breaks down to provide a palisade layer, rubisco, with more carbon dioxide. Therefore, pores can remain open for a reduced time.